Our next presentation comes from Herantis Pharma. I'll hand over the word to CEO Craig Cook.
Thank you. Delighted to be here. Should we move on to the next slide, please? Next one, sorry. Okay, so by way of brief introduction, Herantis is a biotech company. We're headquartered in Helsinki, and we listed on Nasdaq First North in Finland in 2014, and then subsequently in Sweden, Stockholm in 2019. And over the past year, we've made some important strategic decisions. And as a result, the company now is a pure play central nervous system company as of 2021. Our research focus is on disease modification. So we're looking to stop, slow, or even reverse disease pathology with our neurodegenerative assets. We focused on Parkinson's disease specifically, but also more broadly on neurodegenerative diseases. And I think it's fair to say the data that we're now generating after more than a decade of R&D is starting to yield some compelling and intriguing data as it relates to key components of Parkinson's, including the clinical side of things, the imaging side of treatments, biomarkers, as well as genetics. And I'll touch on each of those as we go through the presentation. Next slide, please. So this is our pipeline. CDNF stands for cerebral dopamine neurotrophic factor. And that's a biological protein. It's a natural protein that occurs naturally in the body. And the natural role of CDNF is to protect neurons. And we're looking to take that natural ability of CDNF and harness it as a treatment for Parkinson's disease. That has completed a phase 1. It's a clinical stage asset that has completed towards the end of last year. And the route of administration there was directly into the inner areas of the brain using quite invasive surgical administration means. Moving forward, we're expanding the administration options to include the intranasal route of administration, so nose to brain, as well as subcutaneous or skin injection route of administration. These are far easier, far more patient-friendly, far more regulator-friendly routes of administration, and the focus of CDNF is to move away from the need for surgery to these administration routes. Then we have XCDNF, and this is a chemically re-engineered version of the parent CDNF. So we've taken the smallest components of CDNF and we've re-engineered them chemically, and I'll touch on that program as well during the course of this presentation. That's currently in preclinical. We are about to select the lead candidate to take forward into further development. Next slide. So during the course of this presentation, I'm going to touch on the key aspects as it relates to CDNF and XCDNF, but specifically the key aspects that are important for Parkinson's disease and Parkinson's treatments, including mechanism of action, safety, biomarkers as they relate to CDNF and XCDNF, genetics, as well as potency. Next slide. So to start off with proteostasis, this is the mechanism of action of CDNF and XCDNF. Next slide, please. Now, proteostasis is a key system in the body that influences the fate of every single protein, from production right through to degradation. And proteins are the building blocks of the body. So it's a key system in the body. And when proteostasis starts to go wrong, that's when we have diseases like Parkinson's disease and other neurodegenerative diseases start to manifest. And CDNF and XCDNF are targeted specifically at proteostasis. We're in great company. There's other big players in this space that are also focused on proteostasis, like Roche, Merck, Biogen, Clinical Labs, and Google. And the difference between what what Herantis is doing and what these other companies are doing is explained on the next slide, please. So this just illustrates proteostasis. I'm not going to go into detail on this other than to say that you will note that there's 4 key areas where CDNF interacts with proteostasis, including the production of the proteins, the folding of the proteins into their active 3-dimensional structure, the degradation or elimination of bad proteins or toxic proteins that are responsible for disease, as well as number 4 there is inflammation outside proteostasis that CDNF interacts with this key system. Now, many of the companies that I mentioned previously, they're doing great work. Roche and Biogen, they're doing great work. They focused on a specific segment of proteostasis. Herantis, we're also doing great, fascinating work, but we focus on the entire system of proteostasis. So we have a different approach to these other companies. Next slide. So that's mechanism of action, proteostasis. Important to remember because that is our core offering. From a safety perspective, the second point, moving on to the next slide. This was a study that completed at the end of last year. It was a safety-focused study. There was an initial 6 months where patients were put onto either placebo or onto drug. There was then an extension 6 months where patients that were on placebo were also put onto drug, and currently the study is in follow-up. On average, the patients in the study were around about 60 years old, and importantly, they had diagnosis of Parkinson's disease for 10 years plus. So that's quite advanced disease for Parkinson's patients. So these were very advanced patients in terms of the loss of dopaminergic neurons in their brain, which is the key pathology in Parkinson's disease. Next slide, please. And we are very pleased to announce that the study met its endpoint and safety for CDNF was established. Most of the adverse events were mild and transient. There was a similar safety profile in the first 6 months versus the second 6 months. There was a similar safety profile between drugs and placebo and different drug dose groups. Importantly, there were no dose-limiting toxicities related to CDNF. And although this was not an efficacy study, because it was the patients were so advanced in terms of their disease, what we can say is that we didn't see any worsening of disease in a disease that you would expect to deteriorate over time. So we're very pleased with the outcome of the phase 1 study. So I've touched on proteostasis, I've touched on safety. Next slide, please. Now I'm going to talk about biomarkers and genetics, and this is where it gets even more interesting, really interesting. Next slide, please. So the study was very intensive, and at several occasions during the course of the study, we took samples to analyze biomarkers from the cerebrospinal fluid, and we recently completed the analysis of the biomarkers, and some very intriguing observations have come out of that analysis. First of all, we see that the biomarkers that we analyzed, and this was a panel of 50 biomarkers relevant to the disease, the biomarkers change in response to CDNF treatment. And those changes in CDNF treatment in the biomarkers are actually correlated with improvements in motor function, as well as in biological imaging signals. So imaging that is done of the brain, and specifically the Parkinson's areas of the brain. We also noticed interesting genetic context, since some of the patients had Parkinson's relevant mutations, LRRK2 and GBA. Those are recognized genetic mutations in patients. And finally, the profiling or the signature that emerged as a result of this analysis very strongly suggests that we are modulating potently the system of proteostasis, which I described earlier in the deck. Next slide, please. So this is looking at a patient on the study where we're pulling together all the aspects of his treatment. We're looking at the genetics, we're looking at the motor score, we're looking at the imaging, and we're looking at the biomarkers. It was a 62-year-old male. He'd had disease duration for 10 years. And in the study, he was put onto the first 6 months, he was put into placebo. And the second 6 months, he was then changed on to CDNF. So the first point to make is that his genotype or his genetics were LOCK2, which is a recognized Parkinson's mutation. Then we looked at the motor score. And the tool that we used in the motor score is something called UPDRS. And the lower the score, the better. So in the first 6 months, we look at the left-hand column of that graphic under number 2. While the patient was on placebo, we see that there's no real improvement, maybe a slight improvement, but overall nothing remarkable. When the patient is transferred onto CDNF, which is the second column, the second 6 months, we see a dramatic improvement in the motor score of this patient as assessed by UPDRS. Then we had a look at the imaging, which is the graphic under number 3. And here, higher is better because we want as much of a signal as possible done through the scan of the brain. In the first 6 months, the patient is on placebo, and we see that signal deteriorating over time. But once the patient is switched onto CDNF in the second 6 months, which is the second column over there, we see that stabilize or potentially even increase slightly as a result potentially of the CDNF treatment. And then we had a look at the final piece of the puzzle, which is the biomarkers, the proteomic profile under point 4 there. And again, it's split into 2 columns. The left-hand side is the first 6 months on placebo. The right-hand side is the second 6 months on CDNF. And red means response. So in the first 6 months, we see there's not a lot going on there. In fact, there's deterioration in many of those biomarkers as they change color into blue and yellow and white. But once the patient is put onto CDNF, we see those biomarkers light up red, which indicates a definite response to the CDNF treatment. So when we pull together the entire picture of this patient's treatment, we see a very intriguing and compelling picture that covers the genetics, the motor score, the imaging, and the biomarker profile. Next slide, please. What about XCDNF? So XCDNF, remember, is the chemically engineered version of CDNF. Next slide. And what we've done here is we have re-engineered CDNF and taken the smallest, most potent components of that, specifically the C-terminal domain, which is the green area you see on the right-hand side of the slide there. And we've re-engineered it to achieve 2 things. One is to cross the blood-brain barrier, and number 2 is to retain the potency of CDNF in protecting neurons. And this is given via a simple skin injection to patients. Next slide. So the first aspect that we had to make sure we could get right with the re-engineering is blood-brain barrier penetration. So here, this is a study in mice that demonstrates exactly that. The y-axis is the concentration of XCDNF, and the x-axis is the time after administration. And what we're seeing here is the blue trace is the concentration in plasma after the injection, so nothing remarkable there. You expect it to go up and down. The red trace is the proportion of XCDNF that has crossed into the blood-brain barrier. And what we see there is an impressively significant proportion of XCDNF has actually crossed into the blood-brain barrier. About 20% has managed to cross into the blood-brain barrier and once into the brain, meaning it's crossed the blood-brain barrier. And once it's in the brain, it sticks around for a long time. And as long as it's there, it can do its job, what it's meant to do on the disease areas of Parkinson's. So this is very impressive proportion that has actually crossed into the brain. Typically, you would expect less than 1% to be able to cross the brain. That wouldn't be unusual. Here we're managing to get 20% across the blood-brain barrier. So a big tick in the box for the blood-brain barrier penetration. Next slide. What about the potency? So this is a study in animals again that we did, and these animals were induced with Parkinson's type disease. And then they were split into 2 groups, one that didn't get any treatment and another that did get XCDNF treatment. On the left-hand side, we're looking at the survival of dopamine neurons. So on the y-axis, we have the number of dopamine neurons surviving. So those are the key nerve cells in Parkinson's that need to be preserved. And on the x-axis, we see different dose groups. The gray column here is control animals. So those are animals that weren't induced with disease. They're just there to control. So you'd expect 100% of their neurons to survive. But then the colored columns to the right are animals that were induced with Parkinson's disease. The red group is animals that were induced with Parkinson's disease, but did not get treatment. And we see they lose about 50% of their neurons during the course of the study. The blue groups are animals that did get treatment with XCDNF. And for example, if we look at the dark blue group, XCDNF protected 50% more than the neurons compared to the vehicle group, the animals that didn't get treatment. And that's a very significant result. This is only 4 weeks of treatment. These animals only got 4 doses, 1 dose per week. So it was statistically significant as well as observationally significant, the difference in the number of neurons that survived in the CDNF group, XCDNF group, compared to vehicle. Then we look at the graph on the right-hand side. And here we're looking at alpha-synuclein. Alpha-synuclein is a bad protein. That's one of the proteins that cause pathology in Parkinson's and closely linked with Parkinson's. So here, the lower the better. You want this to be low. On the y-axis, we've got the concentration of alpha-synuclein, and on the x-axis, again, we have the different groups. The control group, normal animals, no disease, you'd expect it to be low. The vehicle group, the alpha-synuclein levels in the vehicle group are very high, as would be intended by this model. But the animals that got the XCDNF treatment, we see that the alpha-synuclein levels are dramatically reduced, almost below supranormal, because they go below the control animals. So a very significant effect of XCDNF on alpha-synuclein aggregates. So again, a big tick in the potency box for XCDNF. Next slide. I won't spend a lot of time on this slide in the interest of time, other than to say this is looking specifically at markers of proteostasis. So remember, proteostasis is a key component, key system that goes wrong in Parkinson's disease. And this is looking at specifically at markers of disease as it relates to proteostasis. And these markers typically increase as it relates to what is called the unfolded protein response. So the intention here is with treatment, you want to get these markers as low as possible. So across the board over here, if we look at the groups, we see that in the treatment groups of animals that received XCDNF, up to 85, 90% reduction in the markers as it relates to proteostasis. So definite evidence that we're having an effect on proteostasis. Next slide, please. So in summary, next slide. This is an overview of our projects and our pipeline. CDNF, as I mentioned, is a clinical stage asset. It's completed phase 1. We are now looping back to the nose to brain administration and the subcutaneous administration of CDNF. XCDNF, we're at the point where we're just about to select the lead. And on the right-hand side, we have some of the news flow that we expect to generate over the next 12 to 18 months, which mimics the activities on the left-hand side of the slide. And we expect a rich news flow over the next 12 to 18 months. Next slide. So in summary, it's been quite a lot to get through, but there's quite a lot to tell about CDNF and XCDNF. This slide sort of pulls out the key aspects of both assets, specifically the multimodal mechanism of action that we know restores proteostasis and reduces neuroinflammation, which are both key pathologies in Parkinson's. So that's clear evidence of target engagement. For CDNF, we know it's safe. We know that there's a biomarker response. We know that that biomarker response correlates with clinical and imaging findings. There's also this genetic context that we see in some of the patients. And also there's no worsening of disease over 12 months of treatment. And for XCDNF, we see that chemical engineering has been successful because from a simple skin injection, we know that we can cross the blood-brain barrier, we know that we can protect the neurons, we know that we can get rid of the bad proteins that are related to the pathology of Parkinson's, and we know that we can reduce neuroinflammation. So I hope that's given you a good overview of Herantis and our assets in Parkinson's disease. As I said, there was a lot to get through, but I hope that the key aspects that I wanted to communicate, you remember those, because there's certainly a lot that we are building on as we move the assets forward. Thank you.
Great, thank you very much for that presentation, Craig. A few questions. First, a question on the finances. What can you say about your current cash position and your expected funding needs looking forward in the next few kind of near to medium term?
So so last year we raised approximately 15 million in total, 5 million roughly earlier on in the year, and then 7 million earlier on in the year, and then 8 million in December. We now have a runway through to early 2022, and clearly we need to make sure that we address the funding needs of the company moving forward. But the runway right now takes us into early 2022.
Okay, great. And you spent some time talking about proteostasis, the kind of the core mechanism behind CDNF and XCDNF, and you mentioned some of the other actors within that field. Obviously, there are more companies beyond that, of course, Denali and Humanity, and etc. How should one think about the fact that so many companies seem interested in the proteostasis space, specifically within Parkinson's disease and neurodegenerative disease? Is this competition, or is it validation for you kind of being in the right space?
I think it's certainly a validation. These companies could be collaborators, they could be partners at some point. It's a highly active area of research, a highly promising area of research, and the deals that are being done in that area, for example, with Prothena and Roche, or Merck and Biogen, are billion-dollar deals. And as I mentioned, those companies are focused on specific components of the entire proteostasis system and doing great work. Whereas with Herantis, we focused on we have a different approach because we focused on the entire proteostasis system with CDNF. So in a great space, very promising area, and a solid foundation to build on.
Okay. And you spoke about the formulation work for CDNF primarily. Just looking back, can you kind of just take us through the rationale for switching towards non-invasive routes of administration, and I guess also give some flavor on to what extent you believe you'll be able to leverage learnings and progress that you made with that clinical program into the future clinical program for CDNF.
Yeah, so the one of the biggest drivers to move away from the surgery is that as long as we needed surgery to administer CDNF to patients, we were always going to be limited to late stage patients, very advanced disease. Whereas it's important for CDNF and for any therapy really to get in as early as possible, so that the dopamine neuron, it may be deteriorating, it may be declining, but that's when CDNF can get in and turn things around. Once the neuron is lost, then no therapy, not even CDNF, is going to be able to replenish those neurons. So it's key for us to be able to get into earlier patients. With these new administration routes, that's exactly what we are able to do. We expand the target population to a much bigger population, a much earlier population, whereas with the invasive surgical device, we would always have been boxed into late stage patients. And it's so that was the key driver. Other drivers or other factors that were taken into consideration was the regulatory perspective and the hurdles to go through that, the burden that it places on patients and hospitals, because it's invasive, it needs hospital time. It's an intense period and observation for patients, And also to complete the picture, potential partners interested in the program, the invasive surgical route was an issue for them. So I think the fact that we moved away from the need for surgery addresses all of those components or those concerns that maybe existed with a surgical device as long as it was part of the formula. Now we're backing the drug on its own without the need for any surgery.
Okay, great. We're all out of time. Thank you very much for an interesting presentation, Craig.
Thank you. Thank you. Delighted to be here.